Okay, good morning, everyone. So, for today's lecture, I wanted to cover more about um, um, like addiction and how it affects brain. And um, I have been interested in internet gaming disorder. Um, Not that um, I'm addicted or anything, but just as a interest that there are many different kinds of addiction. Um, like alcohol, like marijuana, cocaine, many others. And then now there is, like screen time, right? And then internet gaming disorder. Um, and then I wanted to cover what are the key findings across different disorders, what are their similarity and differences, and by using these, can we get some insights? So, reward processing is like the engine of motivation, and when the system breaks down, we often see problems in both addiction and affective disorders. Like, does anyone know what affective disorders are? Hmm? Yeah, depre anxiety and things like that, like general anxiety disorder and so on. So in addiction, whether it's gaming, social media, or substances, the brain control circuit weakens and behavior becomes more compulsive. And in anxiety and depression, we see um, disruptions in the same frontostriatal areas, which may actually predispose people to compulsive behavior. What's interesting is that across these conditions, the same brain networks, especially the striatum and prefrontal cortex, are involved. Uh, right? So um, this is just showing how in these cases, there is the limbic, associative, and motor system, and the frontal striatum is associated in all the disorders. So the first thing is to look at um, across different addiction disorders, right? And like in here, we are looking at reward anticipation versus reward outcome in addiction. And this is reward anticipation, reward outcome. And the distinction between anticipation and outcome is crucial because anticipation deficits, which is hypoactivity, may reflect diminished motivation for non-drug rewards. In outcome phases, increased activation in, let's say, substance abuse disorders may reflect exaggerated response when reward is surprisingly or unpredictable, which would be like a reinforced learning error. In gambling disorder, the dorsal striatum hypoactivation may reflect different habitual learning process, right? Right. So this is all addiction versus control. This is substance addiction versus control. This is gaming addiction versus control, right? And this is for reward anticipation. and this is reward outcome, right? So there are, um, while there are some differences between these, um, but there seems to be more difference in the outcome, right? So during reward outcome, substance use disorders show increased ventral striatal activation, while gambling disorders show decreased dorsal striatal activation. Right? And these patterns support both reward deficiency and learning deficiency hypothesis of addiction. And so the first thing we wanted to do was look at neuroimaging research is increasingly aiming to identify biomarkers predictive of relapse in substance dependent individual, and to see whether we could use resting state in addition to what was done like structural imaging or functional imaging. And the finding typically has been for lower activation in control region like dorsal PFC and altered connectivity activity in insula with DACC network predicts poor outcome. And that these translational direction underscores that neural circuits we have discussed are not only theoretical but clinically relevant. And the final hope is that maybe one could use TMS and other measures to change someone's, behavior. Again, like in this, the idea is that maybe by better understanding these disorder, one can have a mechanistic understanding, and we can use these brain circuits to anticipate treatment response or risk of relapse, right? And these studies are showing that patients with lower prefrontal cortex or higher insulary activity are more likely to relapse. So as I mentioned, we want to use both resting state and task-based fMRI, specifically in this case, naturalistic paradigm, to better understand the functional alterations during reward processing, salience and control, right? So because we are doing, want to study addiction, that maybe during movies, we can present scenarios which may trigger one group versus the other group. Also, to use structural MRI, and then especially use VBM in conjunction with multivariate structural ICA, right? So the structural ICA is very similar to the way we do functional ICA, right? So each time point in functional is a data point, right, across time. Can anyone tell what does each data point in structural ICA represent? A different subject, right? So if you have 200 subjects, so you can make I wouldn't call it a time series, but a series of data points where each data point represents a different subject, right? So if you have, after they have been transformed into MNI space, we can look at what is the covariance pattern and use it to identify specific patterns in the brain. Also, we can use meta-analysis, like two commonly used one. And meta-analysis is used to get like if there are, let's say, 200 task activation studies that have been done in Alzheimer's, right? And I wanted to know which brain regions get activated in task activation study in Alzheimer's, then I can do a meta-analysis, right? So in meta-analysis, what you do is you take the locations and represent in a brain for the first study, then you do it for the second study, third study, all the 500 study, and then you find where the large clusters are, right? So then you say that, okay, when you do task activation, these regions are typically activated, right? Of course, you could go one step further, and you could do, let's say, which region get activated for working memory, and then get these maps, or you could do motor and so on. So you could be more specific. And once you have these measures, you can do multivariate or structural equation modeling, right? Like in time series, we do model. So this, you have like 3 regions where you can say this is the connection between, like, A is connected to B, B is connected to C, and C is to A, and what is the relative strength. So it will give you some idea as to that, okay, these are connected and this is the way they are connected, right? So the first study looks at how internet gaming disorder symptoms relate to changes in the striatum, a key part of the brain's reward system. And what we found was that people with higher IGD, which is internet gaming disorder, they have severity that showed reduced striatal volume, especially in the right caudate. Also, these patterns were replicated across male and female, suggesting that it is a robust marker rather than a gender-specific one. Further, using mediation analysis, we can show that, so this is called Internet Gaming Disorder Scale, short form. This is one scale, and this is Leibovitz, another scale to look at like anxiety levels. So you can look at specific brain regions and see how these 3 are connected, right? So as I said, the main finding are reduced striatal volume, especially in the right caudate, correlate with IGD severity. And this reduction is replicable across genders, suggest vulnerability marker. And these are striatal variation mediate relationship between IGD symptoms and Leibovitz social anxiety scale, right? So between 2 different measures, there are link between them, right? And so this is using open data with subject with cocaine disorders, and in this case we examined the structural brain changes and how it linked to long-term cocaine use, because this in the subject they had asked the frequency of use as well as the duration of use, right? And what we found was that there is widespread gray matter reduction, mostly in the frontal limbic and striatal thalamic network, right? Which are key to like motivation and control. Again, using surface-based morphometry, we also identified covariant brain volume pattern tied to dopaminergic pathways here, and how they are correlated to different one. And this was consistent with the drug's neurochemical profile. And so, right, so this is just showing, like, this is the dosage, and this is how the gray matter volume got affected with the dose used as well as consumption for how long you have been using. Both are certainly effect. In this meta-analysis, what we did was we wanted to find common and separable neural alteration in substance abuse disorder using a coordinate-based meta-analysis for functional neuroimaging study, because there are many neuroimaging study. And so we wanted to do a meta-analysis, and what we found was that cocaine and alcohol involve frontal limbic alteration, while nicotine impacts insula thalamic system, right? So this is showing if we only did 2 clusters, these are the 2 regions that would be that would show. If we did 3 clusters, right, and so on. So these are the regions which showed significant activity across different different fMRI studies, right? However, in contrast, if one was using only nicotine, it primarily affects only the insula and thalamus, right? Uh, I believe they are between 25 and 40 some years. So they were not very young, nor very old. So they were in between. So then after we found some differences, we wanted to know that, you know, whether there is some similarity between obsessive compulsive disorder and people with substance use disorder, right? That maybe could they share some common areas. So so here we compared with like substance abuse disorders with OCD to see like brain overlap, right? So the first is looking at anatomical overlap, and the main region is that decreased inferior frontal gyrus across both SUD and OCD suggests deficit in cognitive control, while for functional dysregulation across SUD, IGD, and OCD, meta-analysis reveal a common anterior insula dorsal striatal alteration supporting a compulsivity-related neurocircuitry. Again, for the structural one, we didn't use IGD because IGD is a very fairly new disorder, if you will, right? So there is not many studies that has been done, but in functional imaging there has been done. So these were the main finding, right? This final meta-analysis looked at shared functional dysregulation across IGD, SUD, and OCD, right? And what we found is an overlap in the cingulofrontal regions especially involved in reward anticipation and executive control. And essentially, these disorders share a breakdown in the brain's ability to balance motivation and cognitive regulation. So these are like, in all these cases, we see a difference in the executive function. So we found structural and functional meta-analysis revealed converging alteration in the frontolimbic and cinguliostriatal circuits, impaired reward anticipation and deficit cognitive control emerge as shared mechanism across SUD, IGD, and OCD. And this evidence supports a community compulsivity related neurocircuitry linking emotion, motivation, and executive regulation. And this points towards a unified model of brain network dysregulation and underlying both addictive and compulsive behavior. Right? So then we wanted to focus specifically on addiction. And this is a study which shows gray matter reduction across affective and disorder. This is a review paper, and by performing large-scale meta-analysis, they found overlapping gray matter density reduction in the VMPFC, VACC, and insula across major depression, alcohol use disorder, and schizophrenia. And that structural deficit point to a common neural substrate underlying affective, addictive, and psychotic spectra. And these brain regions are involved in emotion regulation, value representation, and interoceptive awareness, right? And they listed these regions suggesting similarity across them. Again, like in all these studies, like VMPFC, ACC, the insula, they're all seem to be involved, right? So then we wanted to ask, what about reward relationship with affective disorder, and will it potentially lead to addictive behavior? So up to this point, we have seen how reward dysregulation links addiction and compulsion. But what happens when we look at affective disorders like depression or anxiety through the same lens, right? So in many cases, reduced reward sensitivity or blunted reward anticipation appears early, long before addictive behavior emerge, right? So this raises a key question that could dysfunctional reward processing in affective disorder act as a gateway mechanism predisposing individual to addiction, right? So based on someone's structural and resting state data, can we in some way predict people who are predisposed to this kind of disorders, right? So this is whole brain meta-analysis results of fMRI studies in unipolar depression, right? So this is general reward, natural reward outcome. This is monetary reward outcome, monetary reward anticipation. And basically what it shows is that across fMRI study, there is reduced activation in core reward regions, the putamen, the caudate, subgenual ACC during both monetary as well as natural reward tasks. and that these regions are part of the mesolimbic reward system, which normally drive motivation and um, positive reinforcement. And especially in depression, these systems become underactive, or they don't respond as much. And so in this case, like, we want to find not just the regions, but to see if there are brain networks which are getting affected by these, right? Further, um, altered reward processing was found to be integral to the pathophysiology of depression across the lifespan. And so we looked at how reward processing differs between adolescents and adults with depression, right? And in adolescent, the main alteration appears in the mid-cingulate cortex, a region involved with social behavior and social reward-based decision making. And in adult, the main region shows up in the putamen, which leads to, which is associated with anhedonia and habitual rather than reward-oriented behavior. So does anyone know what anhedonia is? Pleasure, right, or pain even, right? So these are, so we find that between young adolescent and adult, there is a difference, right? So then we wanted to combine data from major depression, schizophrenia, and alcohol disorder to identify like shared gray matter alteration. So despite their clinical differences, these disorders show overlapping reduction in key brain region, right? So this is depression, schizophrenia, and alcohol use disorder. And these are like the common regions. And so the same region, like VACC, VMPFC, the insula, and the superior temporal gyrus are all involved, are all common to all these disorders. So then what we wanted to do was we want to come up with a unified framework, right? So we know that there is a shared gray matter reduction in VMPFC, across affective, addictive, and psychotic disorder, the converging dysfunction in reward, emotion, and executive control circuit, and that there are disorder-specific expression, and depression has blunted reward response, addiction has hyperactive reward drive, schizophrenia has disrupted motivational control. And so all this point to a common diagnostic pathway linking compulsivity and emotional dysregulation. And therefore, we think this supports a dimensional mode of psychopathology aligned with precision psychiatry, where we can use all these measures to perhaps provide response based on individual subject. And so in this case, we used not just resting state, but also a task, a paradigm, right? So in this study, like moving beyond reward-based paradigm, we wanted to explore social and emotional dimension of internet gaming, right? And so we used a naturalistic socio-economical socio-inclusion exclusion task to simulate real life social interaction during fMRI scanning. So we are showing like inclusion task where like 4 or 5 people are talking, and the exclusion task is where one person is sitting at one corner and the other people are chatting, right? And so what we found was, compared to casual gamers, individual with higher gaming severity showed reduced activation in the amygdala and superior temporal gyrus, regions critical for emotional regulation and a social understanding, right? Altered, so this is showing altered emotional regulation and social cognition, and that this activation level, brain activation level, correlated with the IGD score, right? This is again activation level, right, for exclusion stimulus, for inclusion stimulus, and exclusion versus inclusion stimulus also we saw, right? And so across all our finding, one keeps emerging, that is frontostriatal dysfunction lies at the heart of both behavioral and substance abuse disruption, and that this disruption reflects a loss of top-down control from the prefrontal cortex over striatal reward circuit, leading to compulsivity and habitual behavior, right? And that the dorsal striatum contributes to habit formation and reward cue sensitivity, while the anterior insula integrates internal states and salient signal that drive craving, right? So these are, so again, the point is that we can use not just resting state, but VBM, the clinical measures, as well as, you know, in this case, naturalistic paradigm, to look at very specific disorder and try to come up with a framework. So what we have shown is that the reward system, the mesocorticolimbic circuit, including the ventral striatum, caudate, putamen, ACC, MPFC, is a shared neurobiological substrate across behavior and substance addiction and affective disorder. Both behavioral and substance abuse overlap in neural phenotype, warranting unified transdiagnostic model, right? So we should highlight targeted network, we should identify vulnerability and resilient factor, and then hopefully this can help us with clinical diagnosis, and the hope is that with treatment we can also do, right? So we are working like mapping structural changes based on symptom severity, looking at large-scale topological properties, and searching for intervention, both traditional as well as modern. So what I mean by that is that if there is some drug which can be used, like medicine which can be given to this, maybe these methods can be used to assess whether the subject is improving or not. The other option would be to use TMS and other measures at the sites that I mentioned, right? Also, most of this work was done by Ben Kluger-Brown. So he's the biggest Shah Rukh Khan fan I know. So I told him that if there is anyone who knows Shah Rukh Khan, they will be sure to put the two in touch. So if anyone knows, let me know. Okay, so the next thing I want to cover is some of the limitation, right? So this is, so I presented this to show, or maybe to convey that, what we are trying to do is maybe ask a problem, and for that problem, if resting state can be used to answer the problem, that is good. But if it cannot, it cannot, right? But it doesn't mean that we should only study resting state and exclude everything else, right? So if you want to do, let's say, difference in autism, and you want to use only resting state, maybe you can, right? I would be very happy, but maybe you can also use the anatomy and resting state, right? Or if there is task data, maybe you can use all three and see which factors give rise to this, right? So, so I have mentioned perhaps on the first day that like resting state has many limitations, right? And as I said, like 10 years ago, the editor-in-chief of Neuroimage wrote this like really, I would say, hostile paper, but maybe now when I read it, it's not so stressful. And the main point was that what does resting state mean, right? Since you don't really know, as many of you have said, since you don't really know what the subject is doing in the scanner, do you really know what the signal represents, right? And of course, their point was that since most of neuroscience, starting from the day of Hubel and Wiesel, has been driven by like specific stimulus, like flashing checkerboard or different auditory tone. Since this task paradigm has played out so well, why should we even consider anything else, especially resting state, right? So that was one criticism. The other criticism is that with resting state, can we ever tell what is noise, what is signal, right? And like if we could account for all the noise sources, like heart rate, respiration, blood pressure, and so on, would we still be able to tell that, okay, now I have removed everything, this is the resting state signal, and this is just signal, it has no noise? That is not that easy to do, right? So there is always be some uncertainty in what we are measuring, right? And not just uncertainty as to what the person is doing, uncertainty in what is noise and what is signal. There was another paper written by Emily Finn. This paper actually a student of mine sent me like on a Saturday morning and said, don't read it over the weekend, right? But it's like, why would you then send me the paper, right? I had to right away read the paper. So, and they start by saying the so-called resting state, right? It's like someone saying the so-called actor or the so-called researcher, in which participants lie quietly with no particular input or output, has represented a paradigm shift from conventional task-based study in neuroimaging. So I guess they can't just say resting state has been a paradigm shift. They have to like insult and say, right? And then they say our foray into rest was fruitful from both a scientific and methodological perspective. But at this point, how much more can we learn from rest on its own, right? So that is a point, right? In a sense that if you are only going to collect a resting state, like what what question can you ask? Maybe you can ask maybe 10 questions, maybe 20 questions, right? Guess how many papers have been published using resting state? Any guesses? Make a guess. Okay, I wish, Joshua. Now I know why you're so popular. Maybe 20,000. It, it will take another couple of years. Yeah. So there's maybe 15 to 20,000 resting state papers that has been published. But then the question is, now the new papers that are being published, um, can they simply be on resting state, right? Like slowly and slowly, maybe what she is saying is that slowly and slowly it becomes harder and harder to publish a new paper on resting state, right? So I think that is a valid criticism, right? It's like, you use one method and there are so many papers. So if you do another study, right? If one of you was to focus a study on resting state, what would the hypothesis be? Again, if it's only focused on resting state, could you come up with a hypothesis which is just focused on resting state? May not be that easy, right? You would have to think very carefully, you know, like what has been done, what hasn't been done, and then focused on that. Of course, I can say the same thing about working memory, finger tapping, and other cognitive tasks also. But in cognitive tasks, you can tweak it a little bit, right? But in this case, that is their criticism. And then they say um, while rest still dominates in many subfields, data from tasks have empirically demonstrated benefits, as well as the potential to provide insight about the mind in addition to the brain. I argue that we can accelerate progress in human neuroscience by de-emphasizing resting ground in favor of more grounded experiment, including promising integrated design that respect the prominence of self-generated activity while offering enhanced control and interpretability. So what she is saying is that, yeah, maybe, with resting state, it should be used, but in conjunction with specific tasks, right? And by doing that, like this is the example. So before, um, like saying that, okay, this is maybe rest, this is task, this is naturalistic stimulus, and then maybe what should be done is you should use something which has, so now you have some references, right? Like, so with naturalistic task, this is true for all the subjects, right? Because they're watching the same movie. In resting state, this is different for all of them. In task, this is same for all of them, but no one knows what this is, right? And so her point is that maybe that is like we should use both resting state in a way in which we can use resting state as it is, and also task, right? So the does anyone does everyone agree with her? Does anyone see any problem with this? Anyone? So, so you all agree with her? Not a single person to support me. This one? This, yeah, so now they're saying that you have, so the submarine can send signal, right? And you have information about the baseline. So here you have no idea. You just collect the signal and then look at the connectivity pattern. In here, you are only focusing at the surface. Here, you're only looking at what is going on during baseline condition. This is you're only looking at what is happening on the surface. Here, you are looking at the surface, but everyone is watching the same thing, right? The baseline condition is the same for everyone because everyone is watching the movie, right? And in this, like what she is saying is that you can have some way to know about the baseline also by providing some tasks which, or I think what she says is like some like resting state, but also it has task activation. Mm-hmm. Right. Right. Uh, I think uh, I think what she is again. I think what she is saying is that you should have a task, right, where we know what the person is doing, what is happening to the person in the background, as well as what the subject is doing, responding to the task. And while it may not happen exactly at the same time, we can design an experiment where we can find out what the person is like, what is the connectivity pattern at the baseline, and at a certain point, what is happening during a specific task. I don't think so I think she's assuming that the person is paying attention and trying to do the task to the best of their ability, or all the subject, right? Because I understand what you're saying, right? But if you introduce it as a variability, then it will be very difficult to answer either resting state or even task activation. They are paying attention, I think so, right? So I know what you're saying, and for many things I would agree with you, but I would say that if we allow people to change the attention level, it will be very difficult to compare, like to perform. How, maybe Dr. Bhumika, can you help? Like, what he's saying. Like, what would happen to task activation if we don't control for attention? There will be large variability. Right?
So in this case, I think what we compare is passive passive unit is a little different from this, where still little attention is involved. It's just that you have manipulation with attention. When there is manipulation, the variation will be of a certain kind, say something. If you start modulating attention, like if I say that you should keep attention to the right side of the screen, in that case, you are now manipulating attention, so that task related activity will build upon this passive. For tasks. But manipulation is needed. Sometimes you could say predetermined manipulation is needed to happen, which is with the control for during tasks. What exactly?
I understood that how the experiment related variables can be manipulated to their connections or tasks. My concern is that it seems the whole idea is discounting subject related factors. Like what you just decided to have some experiment.
Sure. So even in passive way, like when we do, for example, if I'm doing a task and I'm using scenes or faces, I may want to do a function of a library. So just see where so in that case, what happens for that purpose, which will happen here. The only thing that you not control is that what is it that I'm processing about this. So I may be looking at it that it is moving forward. I may be counting space. It could be different calculations will depend upon what each subject is actually perceiving as, or what is it that the person is processing. Now, in rest, we are just discounting all of that, but we are not saying that there is no thought happening. It's just that there is no controlled manipulation of the thought.
In case he's asking about the individual process, that will reflect in the data.
Yeah.
Individual is not engaging the as others. So that will reflect in the data.
Then I ask if you have a question.
There can be insights.
I would understand how the task, how the perceptive processes are happening, and what I'm trying to highlight is there is only perceptive process that goes apart from the
That's what I said, that when you have visual attention, which is passive, versus which is active, there will be a difference. So in this case, when there is stick viewing, if I'm not asking you to take a decision or give a response, it is passive, but there is visual attention involved. We cannot say that there is no thought or human factor that is part of the process. It is there, but it is not being modulated. I mean, at least it will not be, or it is not in your control as an experiment. But it is possible that, so for example, if somebody is performing, suppose it's an accident, then one person may say like fine graceful, one may say it is forward moving, one may say it is this kind of a dance form. So there could be different ways one can interpret that. And that is what is definitely passive paradigm. So then you start putting a task to it so that you know what is manipulated and what is associated. But there is never a situation where even at passive doing there is no thought in it. There is, so, but it may vary across.
And at the same time, we are having 100% normal typically we have altered processes in pathology. For example, in a schizophrenia, if I'm giving the same similar, they may be totally
But it,
not affect how the data is then will cause
So now that is a very specific situation.
Right.
so will this not make human factors as important as our paradigms to
Yeah.
take them into account, like not just
But in any case, we are not discounting that.
Right. That is true. Also, Rahul, like I understand what you're saying, but even in psychiatric subject, right, when you're designing a paradigm, the goal is that you would like to keep the attention level between the two groups the same. And then the hope is that the other factor, like perception or whatever, is different, and that is what causes the differences in brain activation. Again, it has that has an assumption, right? Because people, when they criticize about resting state and they say that, what do you know? What can you say about what a schizophrenia person is doing during rest? My answer always is that, how do you know what the schizophrenia person is doing during task, right? That I 100% agree. However, even during the task, when you design the task, you design it in a way when the attention is level is the same. Maybe the perception we don't have much control over for most of the task, and that is what leads to difference, in addition to the resting state circuitry, which may be different, which would show differences. Um, we can come back to this one later. So these are some. The other one is, this is also a very nice paper by Christian Beckman um, and like David Cole, Stephen Smith. And Christian Beckman is the person who wrote the first ICA on resting state, right? And in this case, in this paper, I haven't like listed everything, but I'm going to I have it listed later. But in this paper, what they discuss is that when you do ICA, right, just because you get a set of resting state network, how do you validate accepting by using numbers, right? You can test it on 400 subject or 1000 subject or 50 subject and see differences. But what does it mean? But that is the same as with principal component, right? In principal component, you get a set of component, but whether it is biologically or clinically relevant, you don't know, right? These are, you're taking a set of equations and finding a set of solutions which are independent of each other. Are they biological relevant? It looks like they are, but how do we prove, right? So these are, like, I feel, the main limitations, like physiological and motion artifact, like small head motion, breathing, heart rate, scanner drift can mimic or distort connectivity patterns, right? As we discussed, the single most source of noise in fMRI, especially resting state, is head motion, right? And so we always have to be very careful about correcting for head motion, right? And so in addition to head motion, just as an example, when I was showing you about SUD subjects, that, like, one feature of people with SUD is that their heart rate and respiration, breathing pattern varies much more than healthy controls, right? So if one just blindly did connectivity, in one case, the resting state signal would be more noisier than the other group because their respiration and heart rate signal is varying, right? From beat to beat, like it may change more compared to healthy control. So those are issues. The other issue is that let's say you were to scan someone with Parkinson's with tremors. Can you scan, like, yeah, maybe you can scan them, but when you scan them, you have to be very, very careful to look at every single subject and see what the motion parameter is. Otherwise, you won't see, right? You may get a false positive or something which is related to motion and not due to the underlying clinical case. In that case, the challenge is with severity, maybe you will not be able to study those patient population, right? Because they would be moving too much to get data. And like Parkinson's is not just one example. The other example is that now there's a lot of criticism on people doing autism because many of the data bases which have been generated are high functioning autism, right? So there is very few data on low functioning autism, right? So, and high functioning, so then we may be looking at high, so when we say autism versus healthy control, it's not, it typically means high functioning autism versus healthy control. And that may give a different map compared to, let's say, low functioning autism versus healthy control, which we don't really know, right? And in that case, how can we even give anesthesia? Because the moment we give anesthesia, the connectivity pattern will be very different, right? So that is in terms of physiological and motion artifacts. Is everyone clear about this? Yeah. Oh, I was just giving an example that if if a group has too much motion, right? Let's say children, right? We want to study children, healthy children versus autistic children, right? And you want to do the study and you saw that all these kids are moving too much, right? And you say, I'm just going to give them anesthesia and scan them. And let's say the parents agree. Do you think this data set with and without anesthesia, what would be the difference?
With anesthesia, I think it would make less sense for us to resting state. I'm not really sure.
So what?
Because anesthesia with structural imaging, it makes sense for me logically.
Right.
Resting state, it doesn't make much sense.
So why doesn't it make sense?
Because in resting state, you're looking at your brain connection process, etc.
Right.
With no external stimuli. Because I'm not sure, but then I think when you give it anesthesia, there's not even like supplemental processing.
Anyone else? Oh, I had showed figures, maybe Shivani. I had showed figures. What would happen to brain connectivity when you gave anesthesia? Very good. So the connectivity pattern of specific networks would go down, right? And how much it would go down, you like it's not very clear, right? It's subject specific and so on. So first of all, it would be very hard to find subjects who would volunteer to participate where they are given anesthesia. Secondly, you don't really know what would happen to these functional connectivity matrices, but it would most definitely change. So unless you are studying both the groups, you cannot use anesthesia in one group and not in the other. And even when you use anesthesia, all you can say is that between group A and group B, when they were getting when they had gotten anesthesia, this is the difference. Right? So the other thing I hadn't covered much is global signal. I had mentioned how for many analysis, people use like remove the 6 parameters: head motion, then they remove CSF. Many people remove the white matter also. But some people thought, okay, instead of removing CSF and white matter, a easier way would be to take the mean signal and regress it out, right? And when you take the mean signal like across the brain, take the mean for every image, and then you regress it out. So you have the CSF, white matter, and gray matter all together, and then regress out. And so when you do that, the challenge is that it introduces negative correlation in this. So whether to use global signal or not is like a big debate in resting state. And so while many people use, equal number don't use. That is one. The other factor is that we get the data, we process the data, and like from the scripts that I had given you, you can see that there are many different ways in which you can process the data. And whether you use, simple thing like filtering of 0.01 to 0.1 matters, whether you should do filtering or whether you should not do filtering affects the result. So like the pre-processing method affects a lot. Also, how you process the data is also quite variable, right? And for task, there are perhaps only a limited number of ways in which you analyze the data. Like GLM is perhaps the gold standard which is used. I'm not saying that is the perfect way, but in resting state, all of you would know like 8 different methods, right? Seed-based correlation, ICA, REHO, ALFF, FALFF, then what did you say? PCA, anything else? Right, ROI-based analysis, so that's graph theoretical, right? So there are many different ways in which you can process the data and extract the information. And a lot of time, it depends as to what the question is and how you answer, right? Also, as like the third point, many of you have mentioned, right? Like the interpretation of rest and the meaning of rest is somewhat ambiguous. And I think even like what Rahul was saying just now is to do something about that, right? Like rest to me would be quite different than what rest to someone with a clinical disorder would mean, right? I mean, I do agree, but I would again, my point is that um, that may be true, but we are getting the data, so almost from the baseline condition. And therefore, even there are these differences, we are trying to find what these differences are, because these differences are crucial, right? The fact that during rest, where we tell everyone the exact same condition and we are seeing different functional integration, tells us a lot about the underlying condition, right? And again, as I would like to say, like, correlation doesn't mean communication, nor does it mean causality, right? Just because like left motor and right motor are correlated, it doesn't mean these two are connected, right? So that's like like correlation is not the same as causal, right? But I would say that this is also true for task, right? Just because you present a task and you see correlation, we can make the same argument. So these are like number 4. I think this is where task has a real advantage, right? If I if I took you to the scanner and I asked you to tap your finger, or asked you, or we presented you with a visual stimulus, the chances that, let's say, you came back in December and we made you do the same task, the chance is very high that both the temporal change as well as the spatial extent for each and every one of you will be very high, right? The change as well as the region associated with it will be high. And the reason is that when you tap your finger, it's a very robust task, and the signal changes between 5 to 10%, right? In resting state, you know, the whole change across like a 7 to 8 minute scan, there is hardly 2% change, right? So we are looking at the baseline and it is noisy, and we are looking at correlation, right? And the correlation gets affected by noise, right? So if I did resting state finger tapping visual today and you all came back in December, I redid those tasks, most likely the like the overlap between resting state will be lower compared to finger tapping and visual task. But that way, we don't see, not just because of the change in response, but also becomes, other factors become important, right? Whether you had a good breakfast and slept the previous night may play a role for resting state, right? So all these factors play a role. And I have to admit, like in this case, task response has more signal, more reproducibility compared to rest. But um, we can still find significant overlap with resting state across different days, and I think that is what um, like amazes me. Um, and then the final is like clinical utility. And because of some of the reasons I said, we can see differences from between groups. However, like I give this example that, let's say, like they found, right, like a tumor in my motor cortex, right? Um, and Joshua said, I like resting state so much that I only want to do resting state. There is no need to do finger tapping task. I would be very worried. I would say, no, do finger tapping, make me watch whatever movie you want, because I want to map the brain, right? And so um, the clinical utility of fMRI for task has been um, very small. Like there's only 2 or 3 cases, especially like in US, I know, where you can charge the insurance company, right? Only then you think, like only then will clinicians start using it, because otherwise they won't, like if this scan is not going to get reimbursed, then people won't ask for, like people won't, the doctors won't refer the patients to get the scan done, because it would mean that they would have to pay from their own pocket. Okay, so these are some, like again, some of the limitations. So hopefully, okay. So I have a few more slides. So what are, like I think some of the challenges I mentioned, and like hopefully some of you can find the solutions, right? So these are artifact detection and correction, acquisition methods, analysis methods, and applications. So as I said, with artifact detection and correction, there is head motion, physiological, attention, and other parameters, right? And the question is, if we could estimate the head motion while the subject is in the scanner, would it help us? Most certainly, right? You can then scan again if the person moved. Or if there was a way you could design a sequence which was less sensitive to head motion, that would also help, right? So in anatomical imaging, there are some sequences that have been developed which are fairly resistant to head motion. It takes more time, but you can, the way the data is collected, it oversamples the case space, and from that you can identify, like when the person moved, exclude those, and then do the reconstruction, and you can get very good images. So perhaps something like that could be used in functional imaging to make it better. The other thing is like physiological noise sources, right? If we could, by using like different measures, we can measure the heart rate, we can measure the respiration rate. If we could collect that independent of the fMRI signal, that would be another way to clean up the fMRI signal, right? Also, one could do tasks, or low, one could do resting state, where we could try to make sure everyone was paying attention, right? We could make, just as an example, we could have someone look at like a screen, and every time, let's say, the crosshair turned red or something, they just need to press the button, right? It doesn't have to be every few seconds. It could be like every 10 seconds or so, so that um, we, like that way, the attention level for all the subjects are the same, right? And with other parameters, as people are using, with smartphone and other things, you can get many more measures, like how is your, like, not just your heart rate now, but overall, right? In many phones, you can estimate the heart rate and respiration rate. And if you have that information, perhaps that could also be used in these cases to make it better. Then one can also use acquisition methods, right? So in our field, one of the most significant advances in fMRI technology has been the development of like ultra high field scanners, like 3 Tesla and 7 Tesla, right? So when I did my study, it was on a 1.5 Tesla, right? And when I went from 1.5 to 3 Tesla, the signal to noise was like at least 2 times, right? So as technology becomes better, perhaps the resting state signal will be cleaner, right? And maybe I should also say that initially the first few 3 Teslas that was available, it was very noisy, right? Because a 1.5 Tesla was a very well engineered system, because by that time they had already produced thousands of 1.5 Teslas. And I think the 3 Tesla that I had used was maybe the first or the second one in US for that company, right? And slowly, as they made more and more, it became better engineered, and so the stability, the reliability, and other factors became better. So with these things, it became continuously becoming better. So in terms of acquisition method, what can we do, right? So one could be like, maybe you can answer, right? So let's say the data that I have shared with you and showed you was all done with 3 millimeter by 3 millimeter by 3 millimeter, right? What would happen if you had 1 millimeter by 1 millimeter by 1 millimeter? Would you get more activity? Would you get less activity? Would you get better activity? What would happen?
More clear margins between
Good. So maybe the sensory motor cortex, the way we saw, won't look as blurry. It would be very, like, finer details would be present, right? Just as an example, maybe we could use, like, we can see the visual field, right? As I had shown you before. But what could be the challenges we would face by having 1 millimeter by 1 millimeter? Very good. Very, very excellent, right? SNR will reduce significantly. What else? Very good, right? So the time will increase, the SNR will reduce. But let's say we could, so now what has happened, maybe that's what I was trying to say, is that now there have been several sequences that have been developed. These are called accelerated imaging technique, which uses simultaneous multi-slice imaging, right? So you can simultaneously collect multiple slices. So the time increase that would be associated is not there, right? So in the same 2 seconds, you can collect not one by one, but I believe 1.5 by 1.5 by 1.5, right? So you would get, you know, very fine spatial resolution. But some challenges that occur is, I will tell you a few, but you also tell me one or two. One, like some of the challenges that now you have a person's brain, both like functional brain, which has 1.5 spatial resolution, right? So now, before, let's say we were to transform, let's say Rahul's brain into MNI, then Shreyans's brain, and so on, there would not be much difference because each one is 3 by 3 by 3. It's a little smoother, right? But now when you have 1 by 1 by 1, when you transform, you can transform, but there is going to be anatomical differences that will be present, right? So we need better processing strategies. However, once we can account for them, for those changes, the differences that we can find from subject to subject perhaps would be much more meaningful, right? Or even the activation or the resting state map that we get would be, you know, would have less hemodynamic response contribution, right? And we can correct for that. Similarly, as I showed you yesterday, oh, I was to ask you something. Yeah, so the other thing that can happen is that, what do you think would happen with head motion with 1.5 compared to 3? Which one will have more head motion? 1 millimeter, 1 cubic millimeter, or 3 cubic millimeter? Why?
Because in 1 millimeter it takes very less space for the voxel to shift.
To shift, very good, right? And as I had said, when all of us are breathing in and out, that changes, right, the brain by about 0.2 millimeter, right? So with that, you know, there are many challenges that we have to overcome. However, if we get to that point, the images, the connectivity pattern that we would get would be more meaningful, right? And hopefully we can see in more finer detail the connection between, let's say, left and right, and we can find the specific subregions of that. The other one is high temporal resolution, which I covered, right, yesterday, is that if instead of collecting data every 2 seconds, if we could collect data every 100 millisecond, what kind of experiment could you design that you cannot design now to the other, right? Right. So anyone else? Pathik? The question was, if you could collect data every 100 millisecond, you know, for 10 minutes, compared to collecting data every 2 seconds for 10 minutes, what more information can you get when you collect data every 100 millisecond? Very good. Be more specific. You can what is high frequency signal that you can see now that you could not see before? Say. One is respiration. What is very good? Heart rate now you can see, right? I'm going to ask that for the quiz multiple. Okay, Pathik, say something else. So one is you can look at time delay between two regions, right? Second is you can look at high frequency signals um, for the heart rate, right? The other is that we can also see, as I had showed you before, that in some cases, right, we see the we um, as I had showed yesterday, that the heart rate hemodynamic envelope seems to precede the bold signal by about 1.3 second. Like, so these are some. The other thing is that many of the data, in those of you who collect fMRI data, you will see that there is signal dropout, right? And in fMRI, there is a signal dropout, which means that any place where there is, air or CSF, in those regions, the signal will be much lower compared to the brain, right? And there are many regions which are in that area, like orbitofrontal region, which is very difficult to collect. But a lot of times, like, it is a very important function for like many disorders. And so if these could be corrected for with imaging methods, then it will give us even more areas to look at, like, because we can cover more area, and to look at the connectivity pattern. The fourth is non-bold method, right? So bold, what does bold measure, right? Is it a direct measure of neuronal activity? No, good. Hmm? So bold measures what? Convolution of neuronal activity with hemodynamics, right? So it's an indirect measure. Could we think of a more direct measure? If, again, there are several methods like layer fMRI and so on, which are being developed, which is perhaps a little more direct method than bold, right? There is multi-echo method, which is collecting the signal at 2 echoes, and by using that you can correct for more susceptibility related artifact, right? The susceptibility, like the third point, could be somewhat corrected using multi-echo methods, right? So as more correction method are developed, maybe we can look at connectivity in a better way, right? And as non-bold measures are developed, then we can look maybe using methods which may better reflect neuronal activity. So just maybe last year or maybe 2 years, last year there was a paper which was published in Science where the author said that they can measure neuronal activity in animal models, and there was a lot of excitement in the field. And then they later found out that the way the data was collected was not correct. So I don't know what happened. Maybe they had to retract the paper. The fifth point is, again, I haven't covered much, but you can get an idea, right? Like multimodal application, right? Like I showed some, like Nikos Logothetis paper was using electrophysiological signal along with bolt signal. One could do PET imaging just like fMRI, or one could also do PET imaging from a large number of subjects. And as I said, uh, voxel or structural based ICA is done. We could do PET images, right? Subject 1, subject 2, subject 3, and then do an ICA, right? To look at molecular connectivity. There have been now methods in which people are using um, like it's called dynamic PET, right? So just like in fMRI, we collect a series of images. You collect a series of images in PET, and then you do ICA or seed-based to find connectivity pattern. And one way is that, okay, we are seeing connectivity pattern between left and right motor cortex, but if we could find the same pattern during rest in PET in the same subject, then it would mean that, okay, bold is not a direct way. It has hemodynamic influence, but PET is very different, right? It is metabolic activity and therefore more direct compared to fMRI, right? And so this would show that, okay, there are these ways in which we could also collect the data, right? And similarly, EEG, FNIRS, and others. So with analysis method, what analysis method to use, right? Like, should we use ICA? Should we use PCA? There are other methods which are like it's called independent vector analysis. GICA is group ICA. So there are many different flavors that exist. And as these methods are being developed, in ICA, we are trying to find common features across multiple subjects, but there are methods which have been developed, which are focused not just on subject, on pattern which are common across all the subject, but also to find patterns, spatial patterns, which are unique to each subject, right? So then we can analyze the data and then we can say, in this class, the resting state network, which is common across, looks like this, like these are the 20. And then for any specific student, this is the variation, right? So then we can account for both common as well as subject-specific features. So there's a lot of work being done in that area. Similarly, there is many regression-based methods that have been used, but how to use the HRF, right? Should we take a common HRF across for regression-based analysis, or should we use region-specific or voxel-specific HRF across subject? Can also be done, right? There are also the dynamics that people are more and more looking. As I said, in schizophrenia especially, there seems to be the dynamic connectivity is very different compared to healthy control, right? Suggesting, maybe they are not able to keep their attention level the same. It could be that the perception is varies, or it could mean something else, right? So again, by using different analysis method, you can come up with different ways to analyze this, right? The next is, again, I feel this is one of the big challenges, is that can we do single subject connectivity analysis, right? The data that I had showed, we could find, right? But there are many data where we do the connectivity analysis, it's very noisy, right? Especially when you do seed-based, because the seed-based signal has some low frequency fluctuations of interest, but a lot of noise sources. And so when you correlate, these noise sources would correlate with other, and even though the common signal would correlate, it becomes noisy. So I feel for connectivity, especially resting state connectivity, to go to the next level, one has to develop a single subject analysis, right? And, if I could spend all my time not having to worry about students or writing grants, then I would work on this topic to come up with methods where we could just use one scan and we can find the connectivity pattern which is reliable, because that would change, I feel, right? It would be a significant contribution to the field, because then we can do subject by subject analysis and then compare with a group and see where it is similar, where it is different, right? And again, I should say that, there are methods that have been developed that we are working on, which would make these analysis very robust, right? There was also like now, so when I had used, I forget, maybe 5 minutes of resting state scan to find the connectivity pattern, and slowly that 5 minutes became 7 minutes, 7 minutes became 9 minutes, and 9 minutes became 15 minutes, and now there are many data set where they have resting state scan for about an hour, right? And then look at the dynamic properties and then so on, which is very good. The other way would be that, can we do an experiment where we find out, where that curves plateaus, right? And is there any methods we can use to make that like the data point even less, right? So there are different methods that we are working on. Then the question is, as people collect more and more data, what question can we ask? If you had access to, let's say, 10,000 subjects, what question could you ask which would make this very interesting? Anyone? But think of a question that you could ask that you could not ask with 1,000 subjects. Like, what question can you ask? Let's say Joshua and his partner had 1,000 data, you and your partner had 10,000 data. What question can you ask that Joshua cannot ask? No, this, okay, those are very good points, right? Okay, I see what you're saying. So you are saying that um, if one had a clinical population, then it would be useful. But these are healthy subjects. Could you ask a question? And if what question would that be? Okay. I will let you think on that. The other thing is that, this is like AI, right, has taken over the world in the last 2 years or so. Can we use fMRI data for deep learning? You think yes? Uh, but in deep learning, don't you need large sets of data, right? Like I can find from the internet maybe 10,000 photo of cats, right? In different feature, different so on. And then if someone shows me another photo of the cat, I can easily write a program where it can say, okay, it's a cat, right? But all fMRI images look so similar, right? It's like once we transform into MNI space, they'll I don't think you can tell your own brain, right? Even if I showed it to you, right? So in that case, what we have to be a little careful as to what question can we ask, right? So again, I hope, I'm sure it is going to be useful, but I sometimes worry where I see data, not large data set, but small data set showing very high accuracy. So there's a reason I gave this autism data set, right? So I've been working on this data set for a while now. And maybe 7 years ago, the first paper came showing autism and healthy control, and they used machine learning to differentiate between them. The accuracy rate was 93%. Then few years later, another paper came, right? The first one had used maybe 50 subjects. The second one was using 30 subjects, and their accuracy was 98%. But then what happened was someone had access to 200 autistic subjects. Guess what the accuracy rate was? How much? Yeah. And then as the number has kept on increasing, the accuracy rate has gone down. So then the question is, yeah,
I have a question
yeah,
regarding the algorithm. So it's collected from different sites, right?
right,
So if we use it even
right,
as a training data set,
right,
and then we try to use that information in SVM on a testing data set, and we're trying to predict and we get high or low accuracy.
right,
In case of autism or any developmental disorder, depending on the demographics and the sites they are from,
right.
the progression of the disease is very different. So how can you say that we are getting 93% or 98% accuracy is actually accurately diagnosing for another population based on that?
Okay, I agree with what you're saying, but that is the next question. The first question is, even if the data was, very well connected, can you really get that much? So had the people who had done the data analysis before, had the made a mistake in the way, I'm not saying they had like mistake in the assumptions they had made. Was there some kind of data leakage, right? So I feel that we should be a little careful when we are using these kind of methods. I think I'm running out of time. So like patient population is one way to test, right, connectivity, right? One of the first study I did was Tourette syndrome, right, healthy versus Tourette's, and these were people who were having motor tics, right? They cannot help themselves but do like always be doing something with the hand. or scratching. So in that, like we used to show differences in motor connectivity, right? This suggested that, okay, because they have different connectivity pattern, maybe, the resting state is showing something fundamentally different in their connectivity pattern, right? And as I said, if there are disorders which affect white matter, then maybe white matter analysis would show, right? So what other disorder can we use to pursue this, right? So that's another line we can work on. And the other option would be that there are so many um, really, really good animal studies that have been done, and they have used, not just anesthesia, but genetic knockout, mice or rats, like rats with um, uh, like different kinds of um, predisposition to disorders like schizophrenia or autism or Alzheimer's, and then um, try to get a mechanistic understanding of connectivity. So that would be one. And finally, I would like to say that, although I have worked on resting state, I feel like we are trying to study a problem, which is the brain, and resting state is one way, right? I think it is maybe the easiest and the most important way, maybe not, depending on what you're trying to do. But there are other ways also. There is structural, there is DTI, there is task-based, there is, move like naturalistic, and they all have their advantages and disadvantages. But at the same time, you can do resting state in addition to task, right? And vice versa. And I feel that, and they don't have to be mutually exclusive. There is no way, or there's no reason, why you cannot do both task as well as resting. And maybe that is going to provide you more information than either of them, right? And I hope I have showed you different ways, like you can not only get the activation map, but also you can get, what else? You can use the task, use it to calibrate the task activation. All right. Thank you all. Any questions?
If a person sleeps during the
If a person?
sleeps during the stay
Uh-huh.
and versus the same person under sleep, can be found.
Right.
So will there be any?
That's a very good question. So there will be some differences. First of all, um, it depends on which anesthesia you're using, right? That's one. Secondly, right, um, which one do you think will have more head motion, sleeping or anesthetics? Obviously sleeping, right? Um, when you sleep, there are a couple region where the connectivity pattern is affected, but when you get anesthetized, almost all the regions connectivity pattern is affected. Also, the hemodynamic response function is affected. So you have to be careful that way. Okay. Yeah. Yeah. So that's, I mean, I in some way like that someone has gone through all this trouble, but I myself wonder that, can I, like, before I do any study, I am like the first subject, and if I find it hard, then I don't use it. And I myself worry if I can be awake for 1 hour. So yeah, right. I haven't looked in that detail, but I have like I think one can do, as I was mentioning, one can do like a asymptote, right? You can see that, take the first minute, see how the correlation is. 2 minutes, what happens to the correlation or even the resting state networks with ICA, and keep increasing, right? And see at what point there is no longer, right? Yeah.
So by using that task and
Right,
what should be the order of testing?
right.
Should testing be done
I,
before or after?
I before, but depends on the question. Again, that's where it depends on the question, right? So if you did a task um, which required a lot of attention, right? So maybe the example would be, you could do a resting state scan, then do one back, do another resting state scan, then do two back resting state scan. Is there an after effect when you have to do this performance, or does it go back to the baseline immediately, right? So you can ask these questions, right? Yeah.
Based on my experience, if someone feels this repossessed, especially for resting state function,
Right.
are you encouraging him to do this global synchronization?
Um, so I prefer students not to do global signal regression, but there is a person called Marcus Rakel, right? I had referenced him. He's much like he's much more famous. He's a member of the National Academy of Sciences, and he says you should do global signal regression. So, but I would say don't do, because um, first of all, when you average signal with different amount of amplitude, the resulting signal is going to be very different, right? So the gray matter signal is much higher than white matter. The variation is different, right? The CSF is very different. When you average this and regress, in my opinion, what happens is that there are regions from the gray matter which show up as negative, which was not negative before. So you're creating this false correlation pattern.
Some people they're also doing one participant
Right,
multiple sessions of the same but not the same thing, but
right,
they're involving precision.
right, right.
Understand what exactly it is.
Mm, right. So in that case, what they are saying is that you are there are two advantages. One is that you are taking the same subject across multiple days, and therefore any kind of physiological or you know um, parameters that are there are getting cancelled out, and so what you are looking is mostly mostly the signal, and the noise is getting cancelled. That's one. The second one is that now, instead, as I was mentioning, instead of 15 minutes, from that subject you have 1 hour or 2 hour of resting state scan. Yeah, so I have seen um, especially in psychiatry, they are using it a lot. Yeah. Right. So the idea is that those things will cancel out. No, even if it is different, like it makes some assumption, right? So first is that the signal is the same, the noise is the same, similar, right? Then the signal would add up while the noise would cancel. So those are two assumptions, but in practicality, it will be, both will vary. However, because you have more data points, you can do, you can technically treat each session as a different subject and get a group map, right? And then look at, is there anything specific to the one day versus the second day, third day?
But if you're looking at cognitive networks, so
Right,
cognitive effective networks,
right,
the context for the subject or the individual, it must be valid. So, well, suppose 4 days if I'm scanning the same person,
right.
so will that not affect the connectivity patterns?
It, it does, but what they are trying to also do is they are also getting some behavioral measures and trying to correlate. My understand my understanding is that they are, the way they are doing it, they are almost like trying to treat each day as a different subject, and then, but they have like very similar fMRI response. So they want to see if there is any correlation pattern, then it would be significant. That is the goal. And I feel also by having multiple subject, they can have like maybe both cross-sectional as well as longitudinal design.